So today I'm talking with a former research assistant of mine who graduated Calvin in 2011, Sherry Ackerman. Sherry worked with me on a project looking for protein-protein interactions in human cells. Sherry is now a postdoctoral fellow in the Blaney Lab at the Broad Institute of MIT and Harvard. So let's talk a little bit about where you are, what your current research looks like. But could you tell us a little bit about your research journey and how you got here today? Like, what's the common thread in all the work you've done?
Sure. Yeah, and it's really good to be here. It's really fun to connect. I think just as we open, being able to trace this journey back to schools and educational institutions that along the way. It's been a really amazing journey, and I'm really excited to come back and talk about this. Yeah, so where did this all start? In some ways, it started in a 10th grade biology class when I learned how DNA goes to RNA goes to protein, and it literally blew my mind. I thought this is the coolest thing I could possibly imagine in the world, and had to go on and study biology. And then came to Calvin, and Professor Dave Cucci actually was a huge influential force for me. I was in a biology course with him, and he, I was doing normal lab things like pipetting, and I remember him coming up to me and saying, "Hey, it seems like you're kind of good at this. What do you think about doing this for a summer job?" And I had never even considered research. That was not part of my growing up at all. I didn't know anybody who did research. And so when Professor Cucci suggested this to me, I was like, sure? Is that a thing that people can do? And he connected me to, I actually worked at the Venando Research Institute for a summer before working. And yeah, that was amazing. And then had an opportunity to compete for the Beckman Scholars Program at Calvin, and go on to Berkeley. And so I see, these interactions with specific teachers, specific professors, pulling me in the direction of research, helping me see this thing as a real career opportunity. And then when I look at, what is it in science that gets me excited? Why do I do science? It comes back to working on science that is impactful in the world. Looking at the problems around us and understanding that the answers to some of these questions lie in scientific discovery. And that if we understand the world better, if we understand how biology works better, then we have the opportunity to make better therapeutics, better diagnostics, better devices, better technologies that interface with our bodies, with our minds, and all of the learning that is required for making those things is science. And so that's what I want to do with my mind at this point. And I think that's been true for a long time. I actually had a really fun experience of going back and reading some of my original application essays when I was trying to get into grad school, and thinking, man, this is still really true, that I want to use biology and use chemistry, and use engineering to make a difference in the world. And that has not changed. So it was a long answer, but that's
good. That's good. And you're talking about relevant research, and that really fits this week. You published the first author paper just this week on Nature.com. Relates to the pandemic. Can you talk about this work specifically?
Yeah, I'm happy to talk about it. So the thing that motivated this project is, I think, a long-standing need for diagnostics. And that is something that our collaborators actually have been working on for a long time. And so at the Broad Institute, there are a lot of engineers and scientists that all work under the same roof. And this means that the scientists who are looking for new ways of diagnosing viral infections get to hang out with us, the engineers who work on new technology, and new ideas are born from that. So this idea came from talking with Cameron Meervold is the co-first author on this paper. And he's a postdoc in our collaborating lab, Pardis Sabeti's lab. And he and I were sitting in a seminar. We just ran into each other. We were friends from before. And he asked me if we could put viruses into the platform that our lab works on. And we started dreaming about, what would it look like for us to actually have a diagnostic platform that would allow you to diagnose every viral infection that a person could get? What would it look like for every person who goes to the doctor to get a real diagnosis, to know which virus made them sick? Could we design a technology that was comprehensive enough, fast enough, cheap enough that you could imagine testing every single person for every single virus every time they got sick and actually getting that level of, data throughput, sample throughput? Mm-hmm. And so it was a combination of their lab being really application focused in the area of viral diagnostics, and they've worked on, the Ebola outbreaks, the Zika outbreaks. They're really a virus lab, and then our lab being more of a technology development lab, teaming up with them to address this problem. And so we were working on this for a couple of years before the pandemic happened, and then it turned out to be more relevant than we thought it was going to be, or I think than we hope it ever is.
Right.
Yeah.
So you showed at the news conference the other day, you showed some of the experimental data and some of the equipment. Do you have any of that available that you could share with us now?
Sure. I am home, not in the lab, because COVID is real and we're in the middle of it, but I'm happy to talk to you a bit about it. So I brought, can just show you, maybe it's helpful to talk a bit about how the technology works to put some of this in context. So the technology itself is a way of testing many testing many samples for many viruses at the same time. And the way that we do that is by allowing the samples and the assays to associate on their own. So instead of spending all of the energy to make sure that this particular sample gets tested first for flu, then for COVID, then for dengue, we basically separate all of the samples and separate all of the detections that we want to make, and then allow them to associate with each other spontaneously. And we let that random process happen enough times that we would expect to get all of the possible pairings of the samples and the detection mixes, and then just read out all the answers. And we end up getting a lot of data that we don't need, but it turns out that this is a much faster method than doing it in a deterministic fashion. So in order to have all of that happen, we use a technology called microfluidics, which is, as it sounds, a way of moving fluid around on a micro scale. And the way that microfluidics works as a technology is you do this in things that are called chips. So this is one of our smaller chips. It's from the original version of the technology. This chip contains 50,000 wells that we would use to do these experiments. And then this chip is one of the larger ones that we made recently. You can see it's rubbery, it's made out of plastic. The patterning that you can see like there's a darker section and a lighter section. The lighter section is where all of those microwells are. And this chip contains about 170,000 microwells, which are pretty cool. And then I can show you a picture of, let me see if I can get my screen share to work a minute, of what data from a real chip actually looks like. Can you see this?
Yes.
Awesome. So can you see my pointer?
No.
I'm moving my pointer around.
I can see it. Yes.
Okay,
Mhm.
cool.
Mhm.
So this section on the left is a picture of a chip that has been loaded with samples and detection mixes. And each one of these spots is a microwell. And the color of this microwell tells us which sample and which detection mix is inside of that well. So we can allow the samples to fill the wells and allow the detection mixes to fill the wells randomly. We don't need to tell them where to go. We just let them fill wherever they happen to go. And then take a picture and learn where it is that each sample and each detection mix went. And then we wait for an incubation period. And this is what the chip looks like at the end of the experiment. And things that have turned bright green are positive results. So we can use the color. Let's see if I can do this with the shape. So it looks like this one comes from here. So we would use the color of this well to learn which sample and which detection mix are in this well. And then because this well turned green, we know that maybe this patient has influenza or whatever it was, whatever detection mix it was that happens to be in this well. And then this is just a very artsy picture of me holding the one of our chips in front of an array of really pretty droplets data.
So how many different samples are at once? How many different viruses can you scan at once?
Yeah, it depends on the way you set up the experiment. So the technology is really flexible. If you want, you can use it to test a lot of samples for just a couple viruses, or you could use it to test a lot of viruses, but then you're only going to be able to test a couple of samples. And there are a couple of limitations on that. One is the size of the chip. So you can do a total of about 4,500 tests per chip, where one test is a specific sample tested for a specific virus. And so the more viruses you test for, then the fewer samples you're going to be able to put on. The other limitation is the number of color codes. So we have to be able to track all of the different samples and all the different reagents. We have about 1,000 color codes that we can use. And this means that we could imagine testing 1,048 samples against two, like a positive control, positive experiment, negative control experiment. So if you wanted to have like a plus, like yes COVID, no COVID test, and then you would test that against 1,048 samples, that's the most that you could put into the chip. But that limitation is not coming from the size of the chip. That limitation is coming because we only have 1,050 color codes. So it just depends on how you design the experiment, what ends up being the limiting factor in the size.
Well, that's 1,000 experiments. It's still quite.
It's pretty big, yeah, it's true. And just saying there is an edge, right? When you start talking about the size of the American population, then 1,000 is no longer a large number.
That's true.
Yeah,
that's true. Oh. Well, that's quite a ways removed from what you did at Calvin. Can you talk a little bit about how, I guess, wrap up how what you learned at Calvin prepared you for what you're doing today?
Oh man, yeah. So many ways. So, I mean, there is just the basic education, and that is absolutely essential. So all of the things that you learn in introductory molecular biology are still super relevant to what I do. When we think about complementary base pairing and how proteins recognize nucleic acids and cleavage reactions and things like fluorescence and quenching, and I mean all of these things that you would learn in an undergraduate biology and biochemistry chemistry classes, incredibly important. And so I think recognizing that all of science is built on these foundations, that there's no shortcut for this stuff, like you have to learn it and it will stay relevant and you won't forget it because you'll just keep using it. Yeah, it's super practical. I think also like teamwork is a huge part of what I do every single day. The team that I'm working on now, we are working on pushing this technology forward. We want this technology to make it out to people, and that means additional engineering steps. It means coordinating with people in the government who want to see it brought out into the world, people in companies who want to see it brought out into the world. And all of this is really interdisciplinary and involves a lot of really good communication, caring about people, making sure that everyone's interests are being met, people feel motivated. And that sense of I don't know, I don't want to sound too much like an ad for Calvin, but like a liberal arts education, man. It's so important. Like it is not enough to be good at science. You have to be able to communicate that to the world. You have to be able to write about it. You have to be able to talk to people about why the science is relevant and care about those people and figure out what they need and why it is that we're making this technology. Because we're not making this technology just for fun. I mean, it is fun, but we're making it to help people, and that means we have to understand what those people need. Yeah, so that's another aspect. And then I think another thing, and again, I don't know if, yeah, whatever. It's a little bit of an ad for Calvin. I think the aspect of incorporating faith and science has just been really powerful in my life. That Calvin was a place where I learned that my mind and my faith are not at odds. And in fact, they work together. And God has put me in the position I'm in today, not just to be a good scientist, but also to be a Christian. I mean, I have the amazing honor and opportunity of working at an institute like MIT and the Broad, but it's not just to be a scientist there. It's to be a person who cares about other people and is salt and light, and the gift that is my mind is not a gift that I get to just say, oh, this is something for me to enjoy. Like, this is something that blesses the world. And that's having that sense of grounding, that sense of the work that I do is because God has put me here. To God be the glory for this work, is just an amazing way of going through life. And I think Calvin was central to me exploring that and feeling comfortable, owning that, feeling comfortable expressing that. Yeah. So when I look back at where Calvin had a huge impact on me, those things, like rock solid education, loved the education, loved the liberal arts aspects of the education, and really benefited from the faith and learning aspects of Calvin too.
Well, thank you so much for sharing. Thanks for taking the time today.
Yeah.
It's really good to see you.
It's really good to see you too.
Yeah. So keep us informed with whatever new developments you have, and yeah, stay well.
Thank you. You too.
Thank you.